Can you hear me? Yeah, okay, perfect. Well, first of all, thank you for inviting me. It's a real pleasure to be here. And I'm really sorry I couldn't join you through this week because I would have really liked to share with you some more time, and I hope to be able to meet you after. I've been traveling, so my time was limited. Today I'm going to explain to you and show you some of the work that we are doing. So this is work in progress that has been going on for around 10 years now. And it's about modeling globally the marine ecosystems. So, well, you know, I'm here, and this is some of my collaborators, tight collaborators, PhD students, postdocs here at ECM. We have an informal group that we try to do integrated assessment of marine ecosystems. And here you have a little bit the keywords of the things that we work on. Basically marine systems, we do integrative assessments. And what I wanted to tell you is that we work at different scales, from local scales to regional scale to global scale. And what I'm going today, I'm going to show you the work that we do at the global scale. Okay? What I'm aiming to do today is to walk you through first, what is a marine ecosystem model, a MEM? So you'll be hearing me that I'm going to talk about MEMs. So what are MEMs? And why we want to make our life difficult and go and develop these MEMs at the global ocean. So what is our motivation? Then I'm going to introduce one of the MEMs that have been developed for a while, which is EcoOcean, which is the MEM that I developed in collaboration with colleagues from Canada. And that now the development is basically set here in ECM. And then I'm going to show you how we have been using these MEMs to predict what the likely changes, the impacts of climate change will be in the ecosystems globally. And also, we also take these MEMs, they are not unique answers, but they're for us like platforms of, like experimental platforms that are online, that you can turn on and off processes, on and on hypotheses, and assess what may be the impact of turning on and off that process. So I'm going to show you a little bit how we use them. Okay, and I'll bring you finally to some of the conclusions. So what is a MEM? A MEM is basically a complex mechanistic model. So it's a model that tries to represent several ecological processes, but not only ecological. So you can see here at the bottom, we are trying to go, if a minute, okay. What is this? Okay, yeah. Okay, all the way from the capturing processes at the oceanography, biochemistry, hydrology level, all the way to the socio-economic processes that we find in the socio-ecological systems that is the marine environment. So that's the first characteristic. We are transdisciplinary in the processes we want to capture. Another common characteristic is that there are several pressures that are driving these MEMs. It can be climate change, it can be several human activities, it can even be activities or pressures that come from land that arrive to the ocean. And the final characteristic is that the final aim of these MEMs is to provide a series of indicators. It can be time series, it can be means by region, or it can be maps of ecosystem services, how the different ecosystem components and the services that humans take from the ocean are going to change or are changing, okay, currently. The interesting thing, and why I like to develop these MEMs, is that you need to be able to talk with different disciplines. Really, you need to talk with your colleagues from the oceanography department, but all the way to the socioeconomists and even lawyers to understand what is being projected to happen in the ocean. Okay, so if this is complex, why we want to develop these MEMs at the global ocean? Because it's complex enough to do it in a region, why we want to do it at the global ocean? There's several motivations. One is because people say it's impossible, so it's a good motivation. When they tell you you cannot do it, then it's when you start thinking, okay, how am I going to do that? But the more rational ones is, okay, because the ocean, there's several processes that are interconnected. There's these teleconnections that I really like. Ecologists, we don't think about teleconnections, but oceanographers really think about what may happen in one basin of the ocean that may end up impacting another basin of the ocean. They have been working with that for a while. Anthropogenic impacts, quite a lot of them are local or regional, but some of them, as we know with climate change, can be global and are impacting globally differently, but globally our marine ecosystems. And in fact, there has been a large effort in the scientific community to develop these marine ecosystem models at the global level. We have a very inspiring example from the Earth system community, from the modelers that work at the physical, oceanography, and biogeochemical modeling perspective at the global level. And you have seen, for example, what goes into the IPCC reports or the IPBES reports. It's basically based on global, in this case, Earth system models. You probably know this. This is one of the infographics that you find in the 2019 report of the oceans and cryosphere of the IPCC, where, if I manage, where you are going to find this kind of plots where you have, like, part of the plot is a historical projection on what has happened. These are physical dimensions of the ocean, like temperature, ice cover, salinity. You may find oxygen. So you have this HINCAS model. This is a model, right, that HINCAS what may happen. And then you have a projection to the future. This projection, normally you have more than one. So in this case, you have one that is a projection under optimistic conditions of climate change. And then you have one that is about under pessimistic conditions of climate change. And in this case, if you're lucky, you also have some historical observations that you then feed and you tune your models with. So we have been seeing these models for a while about how the environmental, physical, oceanographic conditions are changing. We also have the mandate of the IPBES to go beyond that and to start informing how the biological components, not only in the ocean, but also in the freshwater and the terrestrial components, are going to change with global change. So the mandate is there. And in fact, developing these MEMS, global MEMS, is not something that is recent, but has been going on for a while. There's actually several MEMS that I'll mention to you just after this slide. And this has been possible in the last 10 years because now we have much more computer power to develop these models. We can deal with the data that are teras, gigas of each run of one of these models can be gigas of data. So it's very difficult to, 10 years ago it was impossible. Now it's getting a little bit more feasible even to run them in your offices. These models, although they are all very heterogenic, so because they have been developed in parallel in different labs in the world. So some are developed by size classes. They don't have a species. They have size classes of the organisms in the ocean. Others have functional groups. So, for example, small fish, large fish, invertebrates, benthic invertebrates. Others talk about trophic levels. So they structure the ecosystem by trophic levels, the species groups, life history stages, etc. And all of them have a commonality is that all those nodes or all those ecosystem components are linked. And they are linked by with the interaction that they are having between them. Okay? They are linked, for example, with information on who is whom, decomposition, energy transfer, but the characteristic is that they are linked, all of them are linked. Let me show you, I wanted to show you here, this is from the same IPCC report, 2019, where IPCC already started showing outputs of these global MEMS. And here you see, let me see, yeah, here I can read. This is simulated net primary production, how it will change under two different scenarios of climate change. But then here you have total annual biomass and catch potential. So those figures are made with MEMS, with global MEMS, and including EcoOcean. Okay? These global MEMS and EcoOcean as well, why I started to develop this is because this initiative. FishMIP initiative is for the first time, as I told you, all these MEMS have been developed in parallel, but for the first time there has been an initiative globally that has tried to bring us all together and work collaboratively. So for the first time, all the global and also some of the regional models, we are working together to understand what these global models can project in the future and how far we are from each other. So here you have some of the names of the global models that are now running in this initiative and some of the regional names. And I can say that it has made a difference on this development. It really has pollinated us with new ideas, collaborations, and how we work with that. And what it has done, it has given us a framework to work together, because what we do is we don't present the results of one single model, but we present an ensemble modeling approach, which basically we take the runs of different models as the IPCC is showing you, and we then present the possibilities, right? The rank of answers that our MEMS are giving us, as the Earth System Model community is doing. I'm going to walk you through this figure here. So this is what the Earth System Model community would give us, MEMS, to run our models, okay? So this is a temperature. Here you have the historical projection of how temperature has changed, and then you have different likely projections in the future according to different scenarios of climate change. As I told you, here there's the red one, this RCP 8.5, it would be the most pessimistic one, so if we don't do anything about climate change, we may be in 2100 around here in terms of temperature. If we do things then, and we are optimistic about what may happen in the ocean, we will be around here, okay? And this variability that you see in the runs come from the ensemble models. These are Earth system models, so these are several Earth system models, and here you can see that the spread of the answer is because we are using different mechanistic models that project what may happen for temperature in this case. The blue dot is one that is pessimistic, which is IPSL, and the red dot is one, GFDL, that we know that is more optimistic about what may happen. I'm showing you this because later you're going to see many of these graphs, and then you will be able to follow what the results are. So what we are doing with our MEMS is we are using all this data to run our models, mechanistic models, and understand what may happen on the top of the food web. So with our fishes and our organisms that are, I would say, beyond the phytoplankton, right? So we use the Earth system models to run our models and fuel up to the sharks and to the fisheries. There has been this collaboration, I told you, has brought us to work together and to read some very interesting work that I leave you here to check. But always with this spirit to work in collaboration. So, because what is the problem? We don't really know what are the mechanisms that are really conditioning our ocean. And some models capture or put more emphasis on one mechanism, others other mechanisms. So if we work in this ensemble modeling context, we could be a bit more sure that we are capturing probably some, I would not say truth, but some of the probabilities of the trajectories of change. Okay, so what is EcoOcean? EcoOcean is one of those MEMS, okay? It's a spatial-temporal global MEM. It started as a very regional, large regional MEM, but first we started looking at different, what we call the FAO fishing areas, the 19 FAO fishing areas. Then we modeled the 66 LMEs, large marine ecosystems, and then in 2015 we released the first global model. In 2020 we released the second, and now we are hopefully soon going to release version 3. What it is? So we call it's a modeling complex because you will see the structure. It links different models together. So that's why we call it a complex. And it's basically an integration on a food web because the species and these groups interact, and a niche model, a species distribution model, which is a statistical part of the approach. Okay? So allows species to interact, and then we are also allowing anthropogenic disturbance and climate change to change those interactions and to change the distribution of the species. We have used it for several things, to evaluate fish and fisheries, how fish and fisheries that depend on those will change in the future, diversity and ecosystem changing, ecosystem functioning change in the future, and the impact of climate. And now we are starting to look at the management scenarios. I'll show you at the end. Here you have some specificities that I'm not going to go through them, but I'm very happy to discuss it later with you. It runs for now at 2 resolutions. It's 1 degree resolution and half a degree. Now we are starting to run it at a quarter of a degree and see how long it will take. It's a 2D model, so it basically integrates the water column of the ocean, and the assumption is that the structure of our groups capture indirectly this verticality of the ocean. We can discuss if that is a proper assumption or not. Now it works with around 50 functional groups, which include parameters of around 3,400 individual species. And some of these groups are divided in if they are small, medium, or large. It includes fleets, so 14 fleets per LME. And we start parameterizing it in the 1950s, where we have some proper data on catches and fishing effort. And an important thing is that ecospace, which is a well-known ecosystem or ecological model, is at the core of the computational of the food web interactions that I'll show you later. Okay, so this is a bit the structure. It's for you if you have interest in knowing how it works. We have a static part where we parameterize all the groups and the interactions. Then we have a temporal part where we calibrate with the fishing effort. And finally, we have the spatial-temporal part where we resolve at the level of the map, of the grid, what is happening, and we start running. Okay? And as I told you, here you have, this is what conditions, who interacts with whom, and how those interactions will change in the future. And that is basically the core computations come from a well-known model that it's ecospace that has been there for since the 80s. It's well rooted in ecological theory, and basically has three modes. First, you parameterize. Oh, I'm really challenged with it. Parameterize how things interact, how things eat each other and grow. Okay. And you get matrices of these where you have the interactions of the species and the strength of the interaction. Later, with a system of differential equations, you modify that biomass with time according to changes in mortality because of fishing effort, mortality because of other human events, but also environmental forcing, changes in the environment that causes growth but also causes mortality. And finally, you go in this specialization of your processes. So you go in a 2D grid. What also brings us this ecospace capability is what is called the habitat foraging capacity model, which is a way to incorporate the species distribution models within a food web model. Again, I won't go into the details. I'm very happy to discuss, but basically what it does, it allows us to condition how much a species can eat, growth, and reproduce in a cell according to environmental parameters that you have in that cell per time and well, per space, obviously, because you're in the cell, and per time. It allows you to include as many drivers as you want, so you can totally over-parameterize it if you want it, or be a little bit more clever and decide which ones to use. And what the bottleneck is here is that you need to provide the model with the response functions, how the species will react to different environmental conditions. Okay? What do we use to drive this monster? So we use depth. Obviously, the species will distribute according to depth. Temperature, so we have surface, bottom, depth integrated. We are now starting to work with salinity, oxygen, and ice cover, because especially in some regions of the ocean, are important. We have phytoplankton concentrations of production, and then the historical fishing. This is a first attempt to validate the outputs of the model in 2015 with the total catches. The problem here is that to have global data sets of marine organisms in the ocean that cover the whole spectrum is very difficult. So one data set that we work a lot with is catches, global catches, because it allows us it's a way to at least check if our model is capturing this properly. And here you can see that in the first version of the model, for the functional groups, individual groups, where we're doing quite okay. But if you were looking at the LME scale, of course, the variability between what we have observed and what the model is predicting, well, in some cases is a bit still not optimal, and we are working towards continue improving that. What are the things that we are doing to improve it? First, further include biodiversity in this model. We have done that by parameterizing, so each functional group has a list of potential species, and each species has some information that contributes to the parameters of functional groups. And now we have around 3,400 species included, but also trying to really think how to properly move forward in the way we capture how these species will respond to drivers. And here there are some challenges that we have been facing. For example, when you start predicting where the species may be in the ocean, you can have the potential niche if you use the environmental conditions. But you may end up with predictions of species in areas where they have never been observed before. But the environmental conditions are good for them. So somehow you need to restrict yourself to what you have observed in the past, because you know for sure that those were there. So we have resolved that by using what we call the native ranges, constrain the initial parameterization of the model to where the species have been historically. And after that, we release them, and then they can end up in the tropics, or they can end up in the poles if the conditions, step by step, are good for them. They can move, they can eat, they can reproduce, and they are not fished, they are not eaten. They may end up in other place. But you have to constrain them to the initial conditions. Cell-specific responses to environmental factors have been also something that gave us a lot of headaches, because we were assuming those environmental factors that I was showing you, that they were global relationships between species and environmental drivers. But of course, in a cell, the species change latitudinally and latitudinally and longitudinally. So we have done is introduce that capacity to change the response function to the composition of the species in a cell. Okay? And also, something that gave us quite a lot of headaches was to, and maybe quite logical for you, but it was not logical for us when we started at the regional scale, is that metabolic rates of growth and production change with temperatures. When we are working at a regional scale, all our fishes, all our whales, all our invertebrates were reproducing or were growing with the same rates. But of course, when you go global, the same fish, hake, is not going to be growing the same way in the Mediterranean that in the Atlantic. It's the same fish, has the same parameterization, but it grows differently because the environmental conditions are different. So we managed to somehow capture that. And now what we are doing is testing what are the impacts of turning this on and off. So if we don't know this, is this really important at the end of the day when we predict what the species may be, or is not? It's that we are scientists, we like to get into the nitty-gritty of these things, but is this really important at the end of the day? So we are using that, as I told you, as a test, as a laboratory, I would say electronic laboratory or something, no? that you can try things on and off. Okay, this is a new structure. I won't go through this, but basically you have some things that we modified strongly and others that are new. And now I'm going to show you some results, okay, which I think is the nicest part. The first thing that we did is use our MEMS and pumped through them all this information from the Earth system models. This is easily said, but very difficult to do because, as I told you, the MEMS are very different, all of them. So we had to really standardize things, how we did things, how we went with the inputs, how we went with the outputs. The first thing is, we started talking with the Earth system modelers to see what kind of environmental data they could give us. And here you see two Earth system models, GFDL, EPSL. If you remember the first graph I showed you, remember those dots, the blue dot and the red dot? The red dot was very pessimistic, is this one here. And the blue dot was the optimistic one, is this one here. So here you have, for two Earth system models, different projections of temperature, net primary production, and phytoplankton. And here you have the map. So here you have GFDL. This is the mean temperature in 1999, EPSL. And this would be the mean change if we would be in a scenario of optimistic climate change for the two models, or pessimistic climate change. So you see that the variability is very big. So it depends on which model you're using for your inputs, but also it depends on which scenario of climate change you are assuming. And this is the same for net primary production. You can see here at the end of the day that these models predict the areas in the ocean where primary production will decline or will increase. And of course, depends on what you're using to drive your model, it's going to be different because they are quite significantly different. Okay, so now from our side, from the MEMS, the global MEMS, we had first to agree on which indicators we were going to produce out of these models, because I told you some we are using size, others we are using species, other functional groups. So these are the magic list of indicators that we could all, it's like a minimum common denominator, we could all produce those. And I'm going to be showing you quite a lot these indicators, which is the total consumer carbon biomass. Okay? So the biomass of consumers in general. Here you have results, standardized results of EcoOcean. For example, this is EPSL, the same, okay? The pessimistic, the optimistic Earth system models under the optimistic scenario of climate change and the pessimistic. You see how the projections on how the biomass will change in the ocean, it's substantially different. Okay, and now we go to the ensemble. So if on EcoOcean we add 6 or 5 more marine MEMS, so here different colleagues that are running these models at the global level, we can start getting these results. So those were the first results we got as a community. Okay, let's put it all together, let's see what happens. So here, historically, is what the biomass of organisms, consumers in the ocean has been doing, and those are the projections to the future according to the different scenarios. And of course, the uncertainty comes from running these different MEMS. Okay? If you look at the RCP 8.5, well, you see that you go from not so much change. If you use the EcoOcean, in our case, with GFDL, so the optimist, the one that is not so sensitive, to around 35% of change in the biomass that we project to be in the ocean. And here you see the total consumer biomass, again, comparing the optimistic scenario and the pessimistic. Okay, because our models evolve, but also the Earth system models evolve. Recently, we ran, again, all our models with the new projections of how the environmental conditions which are going to change, which are here. This paper, you'll find all the details. And then we ran our models, and we were able to reduce that uncertainty that is in our ensemble. But the bad news are that we are not closer to here, but we are down here, so that we somehow confirming with the new models and refinement of the assumptions that the biomass in the ocean for consumers is likely going to be reduced in the future. Okay, so now I'm going to walk you through what are we doing with these models, okay? Apart from giving bad news to everybody, what we are trying to do. I told you that we are testing them. We are using them to test our hypothesis on how the ocean works. And I'm not going to show you the results because probably I have been talking too much already, but they are in the paper. But what I can tell you is it matters. It matters quite a lot if you turn on different assumptions on how the ocean, the processes work. It does not matter that much when you look at aggregated indicators, so total biomass. The uncertainty or the change is not so large, but when you start looking at the specific groups and at higher trophy level, it does matter. So, and also it matters when you look at specific regions of the ocean, right? So it's something that we need to take into account. And you have to consider that the IPCC, until FishMIP was working collaboratively, was using one model with one configuration. Okay, so all the projections that we have about what may happen for fish and fisheries until very recently was based on one model and one configuration. So I think we can do better. In terms of climate impact, I'll show you some of the results that we have so far, but because EcoOcean is organized in functional groups or in species groupings, we could look at what may happen for some species. And what we have seen is that the most of the impact is on larger species. So larger species, you see a large decline for climate change. Smaller species, you start seeing mixed signals. So under climate change alone, without fishing, you may have some species that may increase, that are the winners of climate change. But I'm going to show you the comparison of results when you add fisheries to the equation, okay? So basically, this is the third round here that we have, where we had two Earth system models. Now you are big friends, GFDL and EPSL, okay? We had this optimistic and pessimistic climate change scenario, and we ran our models with and without fisheries. So what do we see? When we look at the temporal scale, at the temporal changes only, a time series, the most negative effects that we see when we add fisheries are aggravated when we add, when we had climate change, they are aggravated when we have fisheries. And if you see here, these are aggregated indicators. This is total system biomass, total consumer biomass, biomass of commercial species, biomass of organisms that are larger than 10 centimeters, and biomass that are of organisms that are larger than 30. You can see that the negative change is larger in the higher organisms. So the higher organisms, and it's logical, and the climate change and fisheries do worse. But what is interesting for us is also that there were some organisms that were doing okay under climate change alone, that these positive effects are dampened when you add fisheries. So those effects are lower. You don't see those positive effects, or even they turn from being winners under climate change to being losers under climate change and fisheries. And again, we confirm that this EPSL and RCP 8.5 combination is the worst. So if the world will behave as this model and this scenario of climate change is suggesting, then the scenarios are the worst. Okay, for a spatial change, we also see that there are some areas. So basically, when we run these scenarios without fishing, we see that there is this tropical belt that is a clear loser in terms of climate change, and the temperate and polar areas that may be winning some biomass in the future. And this is also confirmed we call cumulative effects, no? The fishing and impact. But what is interesting is that there's also these Arctic, Antarctic, and North Atlantic that they persist as being winners of change only if the world behaves as EPSL represented. So there is quite uncertainty on what may happen in those areas. Some models are telling us that biomass may change in the polar regions, but other models are telling us that it won't. And the last thing that I want to show you, okay, what happens with the species? I told you that under climate change, there's the clear losers, that are the bigger organisms are the ones that are projected to change most negatively, and they continue. So this is obviously with fishing compared without fishing. They continue to decline. So here you have sharks, large sharks, or large pelagics, etc. And some of the winners, so the smaller ones that I was telling you that they were winning under climate change, are turning into losers. Those were the smaller, so the small pelagics, the small pelagic fish, now starting to really lose under climate change and fisheries. But for me, the most interesting thing is that who are the real winners of this? No? The groups that are projected to increase under climate change and fisheries. And those are two in this simulation. So it would be the small pentapelagic fish and the cephalopods. And they seem to be, and here, winning under this combination of conditions that the future may bring. So I'm going to start concluding. I hope I showed you that we can use these models to start understanding globally what may happen to marine ecosystems, what may happen to biodiversity, to fish and fisheries, and also to the food webs. And what I think is important is to use them as to learn what are the external drivers of change, but also the internal drivers. So what are those processes that make our projections more robust or more uncertain? Because we see that environmental drivers, so the uncertainty that we have in environmental drivers that come from Earth system models, make a difference in our projections of what may happen with the organisms. our internal assumptions on how the ecology in the global ocean works make a difference. And now we are starting to look at, okay, and what about the assumptions in the Earth system models? Because we are now using one realization of the Earth system models, one sea surface temperature, but that also has a lot of uncertainty. So now we are running hundreds of simulations and see if we initialize the Earth system models with different conditions, if that really, we see that that has an effect in our ecological projections. Overall, I would like to believe that the combination of ecological mechanisms project plausible ecological patterns. So what we are seeing is what probably in some regions we are starting to see. For example, we see that these large organisms have been going down, and there's estimates on these declines, and the smaller organisms show mixed responses. In some cases they are increasing, in other cases they are declining. Probably the combination of climate change and fisheries mixes up the signals a lot of climate. So in some areas we may find that some organisms may do one thing or the other. And this is important because that means that those organisms are not good indicators of what's happening in the ocean. So we need to know which are the indicators that are definitely going to tell us about something about the climate, something about the fishes, or are going to be mixing us up when we mix samples. And there's this win-win situation where we are going from larger organisms to cephalopods and smaller bentopelagic organisms that, especially this group, is nowadays not fished, so it's not exploited. So what may happen when this starts to be exploited? This is another question that we may be able to help in the answering with this. Okay, what I'm showing you is work in progress, okay? So don't be too hard on me. We are refining the ecological processes. We are adding different drivers of change. Especially, I'm obsessed with this ice cover thing because it's surprisingly very difficult to obtain data, global data on ice cover and the ecological mechanisms of how species interact with this ice cover. But we'll manage. I'm starting to look at what are the individual contributions of change. Okay, we know salinity, we know temperature, what happens with oxygen, with ice cover, what are the incremental effects of changes in those properties. And the uncertainty analysis, which are fundamental to show all this to policymakers and to then inform IPCC reports. Okay. Our hope is that we are able to use this in a context of cumulative effects, but especially in the development of scenarios of change and spatial management. We are now working on a project that is about nature-based solutions, about how increasing the larger areas of the ocean this is protected, large restoration projects, or sustainable fisheries, how may be able to turn around some of these negative trends that we are seeing. And hopefully we will be able to, we contributed to, for example, this report running our MEMS globally. There is starting to, probably you all know, starting to do there's a lot of discussions of, okay, where we are going to protect this 30% of the ocean in 2030 and what are the best configurations. So these models at the global level are contributing, and I hope they will be more and more relevant. With that, I'll leave you with questions if you have any. Thank you.
So thank you, Marta Coll, for the nice talk. So if